Hello, everyone. As you can see, this lesson is going to be on something referred to as non-disjunction. We'll start by taking a look at this picture that we have here of the chromosomes, and hopefully you'll recall that this picture is referred to as a karyotype. This is a human karyotype where we do have the 23 pairs of chromosomes, 46 chromosomes in total. So we have them arranged from our longest chromosome, chromosome pair number 1, all the way down to the shortest chromosome pair number 22. And then at the very end, at the lower right-hand side, we have the sex chromosome. Chromosomes that we're taking a look at here, the only time that you would see this is when a cell is dividing, because these in fact are duplicated chromosomes. So each one of these in fact did start off as one single chromosome like this, duplicated, they're held together by the centromere. Sometimes it does actually look like I'm showing it here, kind of an X-shaped structure, but in reality, the two halves of it, remember those are referred to as the chromatids or the sister chromatids. In reality, they're, well, kind of sandwiched together as I'm drawing here, and that's what we're taking a look at in the picture. When we do have them arranged in this particular format, what we do have are the chromosomes that come from the father and from the mother that are together. And as it does say down at the bottom here, these are referred to as the homologous chromosomes. So the ones that it's identifying, homologous chromosome pair number 19, and the first ones, again, those would be the homologous chromosome pair number 1. As a reminder also, when we do compare the chromosomes that are from the mother and the father, they actually do have exactly the same genes that are arranged in exactly the same order along the chromosome. But of course, they're not going to be identical. They do have slightly different DNA sequences, and those different versions that we have of the same genes, those of course are referred to as the alleles. So taking a look at this karyotype, what we can do for starters is we can count up the number of chromosomes. And again, if we're talking about a human karyotype, it should be 46 chromosomes. So we can see that, yes, there are 46 chromosomes in total. There are 2 of each chromosome, which is exactly what we want. It is a little bit different in this case when we do get down to the sex chromosomes. Remember that in the case of females, they do have a true homologous pair, 2 X chromosomes, where both of these, again, do have the same genes in the same order. When we do take a look at a male, there is, of course, the Y chromosome. The information that you have on the X chromosome is not the same as on the Y chromosome. The X and the Y do have different genes, completely different genes, and completely different DNA sequences. I also have at the top here that when we're talking about all of those chromosomes except for the sex chromosomes, those are referred to as the autosomal chromosomes or the autosomes. So all of these ones here that I'm circling, it's 44 in total, and those are going to be the autosomal chromosomes, and the other 2, once again, they're just referred to as the sex chromosomes. So with non-disjunction, we are talking about what is taking place during cell division. It can be during either both meiosis that I'm going to focus on or mitosis, but most of the time we are dealing with non-disjunction as it does relate to the process of meiosis. And remember that meiosis is the process whereby we have reduction cell division, where we're taking typically a diploid chromosome number, 2N, adult cell, and we're generating haploid cells. And those haploid cells, of course, are going to be the gametes. So this is also referred to as gametogenesis, more specifically in the male, spermatogenesis, or in females, oogenesis. So this term non-disjunction is referring to what is happening to the chromosomes really when they align during metaphase. They line up along the metaphase or the equatorial plate. And during anaphase, of course, they should be pulled apart and they should go in opposite directions. So if I just show during meiosis 1, just one single homologous pair of chromosomes. So if I use the duplicator, and I'll draw it in sort of an H-shaped structure here, the duplicated chromosome from the male that I'll show in blue, and from the female that I'll show in red. And remember that during meiosis 1, they are going to actually pair up along the equatorial plate. And what should happen from this point is the spindle fibers should attach and they should pull this chromosome to the left and they should pull this one here toward the right. That is the normal case. That's what should happen. So whenever we use this term non-disjunction, that just means that it's not happening properly. So if this is during meiosis 1, as I'm showing in this picture here, then what might happen is that instead of the one that I'm showing in the blue from the father, instead of going in the opposite direction, it actually goes in the same direction as the other homologue, and they end up going to the same pole. So if we do take a look at the pictures that I have here now, the one on the far left-hand side, this is what should happen. This is normal. And if we do take a look at this picture, realize that this is not showing all 46 chromosomes. So in this case, the diploid number, the 2n number, is going to be equal to 4, or we have 2 pairs of chromosomes. What this is showing is the arrangement of the chromosomes when the cell is dividing, and this is going to be during meiosis 1. So the first picture that we're taking a look at here where they are lined up, in this case they're lined up vertically along the equatorial or the metaphase plate. So yes, this one here is going to be in metaphase. Then of course we go into anaphase in the next picture. And what should happen in anaphase is those ones that are paired together. Remember again that during meiosis 1, we do have the pairing up of the duplicated homologous chromosomes. So what should happen is the smaller chromosome should go to the left and the other homologous one should go to the right. And the same thing with the second pair of chromosomes. The one on the left should go to the left and the one on the right should go to the right. So then what we do see at the end of meiosis 1 is that each of the 2 cells that would form would end up with exactly the same number of chromosomes. And then we continue on with meiosis 2. In this case, they line up again along the equatorial or the metaphase plate, only now what we do have is the individual chromatids that are being pulled apart. Once again, once they are pulled apart, we no longer refer to each half of the homologue as a chromatid. Each one is now a chromosome on its own. So with this picture here, what we do see is the one at the top is going to the left, and the other one that lined up on the same side is going to the left, and the other 2 that lined up on the other side are going to the right. That's exactly what should happen, and that's exactly what we see happening in the other cell as well. So again, this is the normal situation, and this should happen with all 23 pairs of chromosomes if we are talking about human cells. The point behind this, of course, because it is meiosis, is to generate the gametes, whereas the cells that we started with in the first place are going to be diploid cells. This is a diploid cell that we do have here. At this point, they are already haploid cells, and then in the end, we end up with haploid cells, which in humans would be the 23 chromosomes in each one of the gametes, the sperm or the egg. So we can see in the picture here that instead of having 2 copies of each chromosome, we only have 1 copy of each chromosome, one of the longer one of the longer and one of the shorter ones, and that's exactly what we do see in all 4 of these. So all 4 of these ones here, the haploid number that we do have is going to be 2. There are 2 chromosomes, which is exactly half of that number that we started with in the first place. So what could go wrong? Well, non-disjunction, again, is the improper separation of the chromosomes. It can take place either during meiosis 1 or during meiosis 2. So the picture in the middle is specifically showing non-disjunction, the improper separation of the chromosomes, right away during meiosis 1. And if this is the case, then every single cell at the end is going to be affected, and they're either going to end up with 1 extra or 1 fewer chromosomes. We do make the assumption that it's just one of the chromosome pairs that's not going to be separating properly. All of the other ones will undergo proper meiosis and proper separation. So we start off with, again, our duplicated chromosomes that are pairing up along the equatorial plate during meiosis 1. We see that the ones at the top, the shorter ones, they're doing what they should do. They're going in opposite directions. But these ones here, for some reason, they're not separating and they're both going in the same direction. So right away, what we're going to see is that going into meiosis 2 already, these ones here are going to have more chromosomes and these ones are going to have less chromosomes. So we continue on with the remainder of meiosis, and what we do end up getting at the bottom is these two cells here are going to end up having an extra chromosome, and the two at the right are going to end up having one less chromosome, less or fewer. The symbols that they use here, so N is the correct number, which would be 23 in humans. N plus 1 means, well, it's the normal one number plus 1 extra. So it's 23 plus 1 that we would get if it were a human cell, and 23 minus 1, or 22 in the case of the other two. So if we do have non-disjunction that is taking place during meiosis 1, then none of the gametes, in fact, are going to end up being normal in terms of the chromosome number. Half of them will have an extra chromosome and half of them will be missing a chromosome. With meiosis 2, non-disjunction during meiosis 2, we see that everything is looking fine the way that they are lined up initially during metaphase 1. Separation is fine as well during anaphase 1. When we get on to metaphase and anaphase 2, though, they line up properly. These ones here are okay. They are going in opposite directions as they should. So this one on the right-hand side, it in fact is going to generate gametes that do have the normal number of chromosomes. But if we take a look at the other one going into the second meiotic division, these two here, the shorter ones are separating okay. This one is not. It ends up going in the same direction. So we end up with this one here that has extra, this one here that has fewer, but we do still get two of them that are going to have the normal number of chromosomes. So when this does become a big deal is when these parents are going to be reproducing. And of course, this is how the gametes are generated. And when they do come together during fertilization, that's when problems are created. So if we say that this is our parent number 1 that we have here, where non-disjunction is taking place, and we have parent number 2 here, where non-disjunction is also taking place. So what are some of the possibilities that we have in terms of what we can end up getting in the eventual zygotes that are produced and the embryo that eventually arises from that? So if we do have this one here, this gamete that is joining together with this one here, now what we're going to have is a total of 48 chromosomes. So if there are 48 chromosomes, remember that 46 is the magical number, and having extra chromosomes is not a good thing. Having fewer chromosomes, that's not a good thing either. And having two extra, this in fact would typically end up being so damaging that is lethal, and that would result in a miscarriage, a spontaneous abortion, very, very on probably during embryonic development. Well, what happens if we take these ones here? This one is missing, this one is missing. Well, this is going to be equally as bad because now there would only be 44 chromosomes in total. So this would also typically end up being lethal. So two extra, two fewer chromosomes, very, very unlikely that that embryo would ever survive at all. What about having just one extra or one fewer chromosome? Well, if we have this as our parent number 1 now, and let's take the one in the middle as our parent number 2, gametes that are generated from them. So if we do have this gamete here that is joining together with this gamete here, now we're going to have a total of 47 chromosomes in total. This is only one extra chromosome, and it does depend a little bit upon which chromosome it is that there is an extra of. Remember that the lower number chromosomes, they are the bigger chromosomes. They have more genetic information, and typically if there is an extra one of those embryos would not survive. But when we get to the higher number chromosomes, the smaller chromosomes, there are some cases where there can be an extra chromosome and they do survive, but typically there is usually something wrong with those individuals. And in most cases as well, then This is also going to end up being lethal. We will take a look at some of the examples of where there is an extra chromosome, and they do actually survive at least until the stage where they could be born. But once again, most of the time, somehow the embryo has a way of kind of counting and keeping track of the number of chromosomes and aborting that pregnancy if there is an extra chromosome. What if there is, well, fewer? So this one here and this one, that would give us a grand total of 45. And again, there are exceptions where individuals do survive, and there are some exceptions as well where they are pretty much indistinguishable from people that do have the correct 46 chromosomes. But just like having one extra chromosome for most of the chromosomes, if you are missing one, this would also end up being so lethal that that embryo would never reach the stage of a fetus or never reach the stage where it would be a successful pregnancy and a baby would be born. So let's take a look at what some of those exceptions might be where there can be an extra or a missing chromosome and those individuals are born. So we'll take a look at a few different karyotypes here. So the one at the upper left-hand side, we can see that we have all of the chromosomes that are lined up once again, starting with the longest chromosome pair number 1 at the upper left-hand side and the sex chromosomes at the lower right-hand side. So one thing we can tell, of course, right away, if there is a Y chromosome, then it is going to be a male. So this will be a karyotype of a male. But if you count up the total number of chromosomes, this does have an extra chromosome. So we would refer to this as 2n plus 1. 2n, of course, is the 46, plus 1 equals 47 chromosomes in total. And if we do take a look at which one is extra, it is the 21st chromosome. Now, the 21st chromosome is one of the smallest chromosomes, so this is one of the rare instances where, yes, there can be an extra one, and these individuals do survive. And not only do they survive, but in this case, they are also fertile. So they themselves can actually undergo in their gonads, the testes in a male, the ovaries in a female, they can undergo gametogenesis and they can produce gametes as well. And some of them will end up having the correct number of chromosomes. Some of them will end up having an extra chromosome. So this particular case here, where there is an extra 21st chromosome, because there are 3 of them, it is referred to as a trisomy. And we just indicate what the number is. So this would be a trisomy 21. But this is also referred to as Down syndrome. So Down syndrome, extra 21st chromosome. And again, this is one of the exceptions where not only do they reach the end of the pregnancy, not only are they born, but they actually do survive and are also able to reproduce. The one that we're taking a look at on the right-hand side, so in this case here, there is no Y chromosome. So this is going to be a female that we're taking a look at here. Count up all of the chromosomes, and once again, we can see that this is a 2n plus 1, or 47 chromosomes, and here it is the 18th. So this is a trisomy 18, and this one here is also referred to as Edward syndrome. Unlike individuals that have Down syndrome, those that are born with Edward syndrome, they do not survive. They would typically only survive for a few weeks after being born. Other ones that do happen, the next one that we have on the left-hand side here, we can see once again there is no Y chromosome. Because there's no Y chromosome, this is going to be a female, but notice that there's no second X chromosome. So this one is a 2n minus 1. Specifically, it is missing the second X chromosome. So this is a female, and she has what is referred to as Turner syndrome. Turner syndrome, missing the X chromosome. So there might be some consequences associated with this, related to the female's reproductive cycle, related to having children. Body proportions might be a little bit different, or they may not. Female with Turner syndrome, it is possible that she may be one of those females that is indistinguishable from a female that does have the X chromosome. So really a very, very broad range in terms of what we do see in individuals with Turner syndrome. And that is also the case to some extent as well with people that do have Down syndrome. The next one that we're taking a look at here on the right-hand side, we can see that there is a Y chromosome. So this is going to be a male that we do have here. In this case, it's an extra 13th chromosome. So this is a trisomy 13, and this one is specifically referred to as Patau syndrome. And this once again would be our 2n plus 1, or the 47 chromosomes in total. The last one that I have on here, this one is showing a male because there is a Y chromosome. Whenever there's a Y chromosome, at least chromosomally speaking anyway, it is going to be a male. But this is a male that has two X chromosomes. So typically what you would see in this male would be feminization of characteristics, including the external genitalia. So this isn't a trisomy, but it is still 47 chromosomes in total. And this one is referred to as Klinefelter syndrome. Klinefelter. So all of this, I've been focusing on non-disjunction that takes place during the formation of the gametes or during meiosis. But just briefly, this can also take place in mitosis as well, and it's referred to as mitotic nondisjunction. It's really exactly the same thing that's taking place. Of course, during mitosis, we don't have any pairing of the chromosomes. They only line up in a direct line along the equatorial or the metaphase plate, as it does have identified here. And just like with meiosis, during meiosis 2 more specifically, what we should have is the microtubules that has less labeled here, but these are spindles or spindle fibers. And what we should have is that 1/2 of each of these chromosomes, or in other words, the chromatids, should be pulled apart and they should go in opposite direction. So these ones here are showing what should happen, and this one is showing the non-disjunction. So depending upon when this takes place, that might determine, well, how many cells within an individual would actually have an odd number of chromosomes. In other words, a number other than 46 chromosomes. But if you keep in mind that we do have, well, maybe 100 trillion cells in our body, and many of these cells are undergoing mitosis, so probably pretty much guaranteed that not every single one of your 100 trillion plus cells actually have 46 chromosomes. Some of them may have gone through a mitotic non-disjunction, which means whenever that happens, some of them will end up with 47 chromosomes and some of them will end up with 45 chromosomes. If this does take place very early in embryonic development, then there may in fact be a significant number of cells that do in fact have the odd number, the 45 or the 47 chromosomes. And depending upon where that has taken place in terms of the differentiation process, there may even be a significant number of tissues or organs that do have the odd number of chromosomes.